Question number 3. Amylase is an enzyme which digests starch. 3.1 is an A1 question, and this question is from the Human Digestive System. Which organs in the human digestive system produce amylase? Tick one box. Different options are given. Liver produces bile juice. Large intestine is not producing any enzyme. Stomach is involved in producing protease enzyme. Salivary gland is producing amylase enzyme. If you look here, which is the correct option, salivary gland, pancreas, and small intestine, these are the organs which produce amylase enzyme. A student investigated the effect of pH on the activity of amylase. The method is described here. Number 1, prepare amylase solution at pH 5, which is an acidic environment. Number 2, mix the amylase solution with starch in a boiling tube. Number 3, remove a drop of amylase starch mixture every 30 seconds and test it for the presence of starch. Number 4, record the time when all the starch have been digested. Number 5, repeat steps 1 to 4 using amylase solution prepared at pH 6 and then at pH 7 and then at pH 8. Student must know the working of amylase enzyme. Amylase is an enzyme which helps to digest the starch. So when all the starch is digested, it means the amylase has digested the starch into the form of simple sugar or glucose, so it can be absorbed in the body. So relation is between the amylase working and the digestion of starch. 3.2 is an AO1 question and it relates to practical activity number 4 in combined science. And this question is from the Human Digestive System. What was the independent variable in the investigation? In this investigation, the pH of the buffer solution or the amylase solution, that is an independent variable. The reason is when the pH is changing in our experiment or in our investigation, the amylase enzymes take time to digest the starch. So we want to see if the pH is changing, what is the impact on the working of amylase solution. The model answer is the pH of amylase solution or the buffer solution, that is an independent variable. The student must know what is an independent variable. Independent variable is a variable which you can change. On changing that variable, the other variables are affected. 3.3 is from the same topic of the human digestive system and it is a knowledge based question, AO1 question, and it relates to practical activity number 3 and 4. Describe how the student would know when all the starch had been digested. For the starch test, we need iodine solution, and iodine solution has a color of brown or orange color. When the starch is present in the food, the color of iodine changes from brown to black or blue color. When there is no more starch present in the food, then the color of iodine will remain orange or brown. Let's see the model answer. When all the starch is digested, there is no change in the color of iodine. So iodine solution has a color of brown or orange color. When it changes from brown to blue or brown to black color, it means the starch was present. When there is no starch is present, the color of iodine will remain brown or orange. So there is no black color in the test. 3.4 is an AO3 question, which is a practical based activity. And the figure number 6 is given here, a graph. The graph is between the pH and the time taken to digest all the starch in minutes. The time taken to digest the starch, it varies with the change in pH. You can see here the graph is moving down and it is going up with the change in pH. So we have to see from here that the pH values is increasing towards right and it is decreasing towards left, and the enzyme working is showing that it is taking less time to digest the starch, and when the pH is increasing, the graph is going up. It means the enzyme is taking more time to digest the starch. We need this figure number 6, to answer our question. What is the question? What was the optimum pH for the amylase enzyme there? Use the figure number 6. The extra information is given here. The best pH value for which the amylase takes less time to digest the starch. If you go here, this is the time when the amylase is taking the least time to digest the starch, and this value is about, if you could draw straight here, it is about 6.8 pH. So the correct answer is 6.8 pH. A scientist did a different investigation. This is the method used. Number one, prepare amylase solution at the optimum pH. Optimum doesn't mean the maximum. Optimum means the best pH on which the working of the enzyme is the best one. Number two, mix the amylase solution with starch in a boiling tube. Number three, measure the concentration of sugar every 10 seconds for 2 minutes. Figure number 7 shows the scientist's results. If you look here, the figure number 7, the time is given in seconds along x-axis. Concentration of sugar in arbitrary units is along y-axis. A graph is given here. From the figure we can see as the time is passing, the working of amylase is increasing and the concentration of sugar in arbitrary units is increasing. When all the starch is finished, then there is no more working of amylase and the graph is getting flat or shallower. 3.5 is a question which is from the Human Digestive System. How much time did it take for amylase to digest all the starch? We can see here the starch is converted into sugar. This is working of amylase enzyme. We see that the graph is not going up anymore. It is getting flat. It's not going up. So it means that this is the point when the graph is getting flat, and this value from the graph is 82. When the working of the amylase stops, means no more starch is present. So that is about 82 is the value. 82 seconds is the time taken by amylase to digest all the starch. And we need to check from the graph. The graph is very clear. Here it is getting flat. It means no more starch is present. The correct answer is 82. Use a graph when the graph is getting flat means the concentration of sugar is not changing anymore. It means that no more starch is present in the food. 3.6 is an AO2 question related to the practical activity number 4. Determine the rate of sugar production per minute at 40 seconds. The rate means any change with respect to time. To understand this question, we need to go back to our previous topic. Figure number 7 is going to help us to understand the question. At 40 seconds, this is a point at 40 seconds, the graph is curved, so we can't find the straight answer. Students are making a mistake. They draw the straight line from here, from 40 seconds, then they move towards left. and try to find any answer here, which is a wrong way of doing it because it is a curved graph. Therefore, the student needs to draw a tangent. At 40, the student need to draw the tangent here. We touch the point 40. Don't try to draw the graph like in this way. Don't try to draw the graph just passing near 40. They need to draw a straight line which touch the point 40 here. Then the student need to pick 2 points which are suitable for the students. Like I picked this point and I picked the second point which are in the blue color. I need to find the gradient. Gradient means the slope. A slope can be found by the way of the formula y2 minus y1 over x2 minus x1. I already elaborated my question to help the students. This value is actually 54. If you see here, straight here, that is 54. And along y-axis, the value is 4.1. I picked this value on the tangent, which has y value is 2.9 and its x value is 26. So 26 and 2.9. I'm going to use these values in my question to find the answer. Like here, I will use it y2 minus y1, that is 4.1 minus y1 2.9, x2 minus x1 54 minus 26. If you're going to solve this question, my answer is 0.42. But this is a change which happens in 1 second. 0.42 is a change which happened in 1 second. I need to find in 1 minute. So I need to time this value by 60 to make it 1 minute. If you time by 60, 0.42 times 60, my answer is 2.5 concentration of sugar in arbitrary units per minute or arbitrary units per minute. So why am I saying this thing? Because my y-axis is actually the concentration of sugar and my x-axis is the time. Let's see the model answer. Let's see the model answer. First, draw the tangent at 40 seconds on the curved graph. Pick 2 points on the tangent and find the slope of the tangent. M is equal to y2 minus y1 over x2 minus x1. Use the values from the graph which I already elaborated on figure number 7. My answer is 0.42 arbitrary units per second. But the question demand is, if you see here, that we need to find the answer per minute. And we know that 1 minute is equal to 60 seconds. So I need to multiply my this value which I got in 1 second by 60 to make it 1 minute. When we multiply, the answer is 2.5 arbitrary units per minute. The answer is 2.5. 3.7 is an AO2 question. Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. Before using the model answer, we need to understand key and lock model of the enzyme. Let's see how this model works. In this question, we must know about lock and key model. This is our enzyme and this is the active site of the enzyme and this is a substrate. The enzyme is going to work on the substrate and it is going to break down the substrate into simple components. But the active site is affected due to change in the pH. It means this substrate is unable to fit here in the active site and the enzyme is unable to work on the substrate. As a result, the substrate is not converted into the simple components because the pH has affected the active site. It is a 6 mark question. It is an AO2 question where the student need to apply his knowledge. Let's see the model answer. Amylase is an enzyme which is a protein nature. According to lock and key model, an enzyme has an active site, which I mentioned here, where a substrate like the starch will fit in. The change of pH other than optimal value will affect the shape of active site of the enzyme. We can see from this diagram, the substrate will not fit in the active site. It can't fit in here. Very high or very low pH will denature the enzyme and will not digest the starch. We can see here if it is a starch molecule, so it can't fit here in the active site, so enzyme cannot work on the starch molecule and can't convert into simple sugar. Therefore, the rate of digestion will decrease. Means enzyme is unable to break the starch into simple molecule, therefore digestion process will slow down.